Ready to start. Welcome to the Research Fest of Parallel Square Technology Institute. PTI is a non-profit research organization aiming to accelerate biomedical research through proteomics. We think that direct measurements of proteins at high scale, high resolution, and high sensitivity hold the key to making fundamental discoveries in biology, to characterizing pathologies, and ultimately contributing to developing more effective therapies. So let me share with you our perspective, why we think proteomics is so potential and has transformative capabilities. About 200 years ago, many people believed in a vital force called elan vital. And this force animated all living things. And then careful experiments demonstrated that instead of elan vital, there were proteins that were performing all of the functions that we associate with life, such as fermentation, biochemical catalysis, and we could isolate, purify, and even crystallize these proteins. And the ability to isolate and purify proteins drove biochemical research for many decades of the past century. This was the mainstay that established many of the fundamental conceptual frameworks that we have in biology. Are biological macromolecules amorphous, or are they molecularly defined? Are protein conformations and structures imprinted by some templates, or are they determined by the primary amino acid sequence? And not only fundamental research, this ability to characterize isolated types of proteins also contributed to improving human lives. The determination of the primary amino acid sequence of insulin ultimately resulted in improved standard of care for millions of people suffering from diabetes. And then in 1980s, a new technology that allowed efficient ionization of large molecules, converting large biomolecules into ions, provided a very significant improvement in the scale at which we could analyze proteins. So that instead of analyzing isolated single type of protein, we could analyze more proteins. In the very memorable words of John Feng, it conferred electrospray wings to molecular elephants. And this ability further increased thanks to more technologies empowering the analysis of these ions by mass spectrometry, such as developments of detectors, the Orbitrap, development of time-of-flight detectors, recently Astro, and technologies that allow to separate and accumulate ions, such as trapped ion mobility spectrometry. All of these technologies gradually increased our ability to analyze proteins at larger scale. And very important to this development were organized efforts by the scientific community to introduce rigorous benchmarks and community standards as part of initiatives such as CIPITAC that ultimately resulted in generating large-scale data sets suitable for clinical research and improving our understanding of diseases. In the meantime, of course, there's another very important set of technologies that have been contributing to biological research, and this is the ability to analyze nucleic acids, particularly to sequence DNA from the first generation of Sanger sequencing to the second generation to the third generation of ion pores. This has been an important contribution driving biomedical research over the last couple of decades, over the last few decades. And because these methods have become so accessible, they're oftentimes used to provide indirect information about proteins as well. And we can easily understand to some extent why those technologies have scaled more readily if we consider the relative complexity of the genome, the transcriptome, and the proteome. In a typical mammalian cell, such as a human fibroblast, there are near thousands, less than a million messenger RNAs. There are billions of protein molecules, billion with a B. So there are many orders of magnitude difference in the complexity, in the dynamic range of proteins, of their modifications, of their interactions. They're so complex that we're yet to identify and characterize all of them. We don't know all of the proteins making up our bodies. We've never been able to identify them. We've never been able to characterize their primary amino acid sequences, not their modifications, not their interactions, not their functions, not their localizations. It's a tremendously, tremendously complex system that is very challenging. It poses a major challenge, but I also think it's a wonderful opportunity for scientific discovery and for improvements of medicine. So, let me tell you why I think measuring proteins directly is important. There are many reasons. I'll share with you a couple. One of those is if we're interested in describing just protein concentrations, protein abundance, what are the regulatory mechanisms that determine the concentrations of proteins, different proteins in your cells, their difference across different cell types and tissues? We find that about half of that is determined from the synthesis side of the equation, and the fully other half is determined by regulation of protein degradation. And if we want to be able to measure and characterize protein degradation, we need to directly analyze proteins. It is very difficult to make these inferences from measuring nucleic acids alone. But even when it comes to the synthesis part of the equation, it turns out that if we want to know what are the primary amino acid sequences of proteins, there are in vivo mechanisms of alternate translation guiding which amino acid is incorporated to which codon that still require direct characterization of proteins, direct measurements of proteins to know what is the proteome that is made in human cells in vivo, as opposed to predicted indirectly from models. And of course, a very, very important reason for direct analysis of proteins is understanding human diseases. We know that for many diseases, the central contributors, the central players, are protein pathologies. Whether these are modified tau proteins and aggregates in neurodegenerative diseases, whether these are aberrant protein kinases contributing to cancer, proteins are central players in diseases. And we need to have the capability to specifically first characterize what are these protein modifications, protein aberrations, and changes associated with the disease. And because human communities are large and complex and heterogeneous, we need to have measurements at sufficiently large scale that can deal with the associated comorbidities and variations across the population. We need measurements that are specific. And this might seem like a simple thing. It has been a very difficult thing to achieve in the world of proteomics, specific measurements. Many of the current technologies struggle still to achieve specificity. We want to know what we are measuring. And then we need technologies that are affordable, that we can really do this for many diseases for the population. Beyond simply identifying associations, we want to transcend the descriptive nature of those and just finding biomarkers. We also want to understand the mechanisms contributing to disease, because the closer we understand the mechanism, the better chance we stand at ultimately developing effective therapeutics. So all these reasons motivate us to think of how we can overcome the substantial challenges of the proteome. And they motivated starting PTI about 2 years ago. So we're a very young organization. We have grown from just a couple of people to about 20 now. And we are very excited today to share with you some of our initial results from developing technologies to enable proteomic analysis, applying those to biological problems that we think are important and impactful, trying to understand mechanisms contributing to disease. But I think as we share with you today our progress, it is worth keeping in mind that everything that we share is work in progress. We have been working on this for 2 years. For some of the projects that we'll share today, we've been working for mere months. But we are nonetheless very excited to share the progress that we've been able to make, because we think it is beneficial not just for us, it is something that we would like to empower others to use in their industry. So how do we approach the formidable problem of scaling up proteomics, of analyzing proteins? We appreciate and embrace all technologies that can help us with this problem. I think there is scope for a lot of different approaches to contribute. But in particular, we think that mass spectrometry is very powerful, and we have a defined strategy of how we can use it. For a long time, mass spectrometry has been well established as being very specific, very versatile, very powerful, but also has some major weaknesses that has prevented it from contributing significantly to biomedical research. Namely, it is relatively difficult to scale, it is relatively expensive, and we want to address those issues. We want to have sensitive methods that can be scaled by orders of magnitude, that can have sufficient sensitivity and be sufficiently affordable so that they can really make an impact in the real world, not just in research and development, but also in helping develop better cures for diseases. So, how are we going to do this? Conceptually, the approach is relatively simple. We want to be able to parallelize data acquisition. The more data points we can acquire per unit time, the higher the throughput, the more scalable the approach. And the way we are doing this is by parallelizing simultaneously in multiple dimensions. One dimension for parallelization is when we analyze peptides or proteins, instead of analyzing one peptide at a time, we can isolate and fragment large number of peptides. Another dimension which we can parallelize is by being able to barcode molecules from different samples, then mix them together and analyze the barcoded molecules at the same time. And if we're able to combine those two axes of parallelization, then we get the product of those axes. We are able to increase the number of data points that we can acquire simultaneously. And this roughly provides a framework that I can use to illustrate at this very high conceptual level, welcome David, some of the technologies that we'll share with you today. So one important thing that you can quickly realize is barcoding. We need to have molecules that allow us to barcode multiple samples so that when we mix them, we can do this analysis in ways that don't compromise the quality of the data. And in fact, today we are going to share with you two exciting advances, exciting for us on that front. One is making new molecules for doing this barcoding, and the other one is a conceptually new approach to multiplexing samples in a new dimension, in the dimension of time, as Jason Dirks is going to share with you. And as you can imagine, acquiring more data simultaneously sounds good. It's not a piece of cake. It also has challenges. So we have much higher density of data points, and we also built in more structure in the data to really solve the challenge of data density and take advantage of the structure in the data that we build. We need new algorithms, and we've made developments in that direction that Kevin McDonald is going to share. So as we scale the data, increase throughput, decrease cost, we are very, very keen to increase confidence and quality in the data sets. This is not just about let's get more data, we can deal with poor quality. No, we want it all. We really would like to have very high quality in sequence determination. As I already mentioned, simply determining amino acid sequence rather than inferring that from nucleic acids is a challenge. So we want to have confidence in determining those sequences and those modifications. And they'll just give you a conceptual flavor of one of the important things that we keep an eye on. So if we want to determine the sequence of this peptide, each letter corresponds to one amino acid, we break it into pieces, and then we can use the pieces to put together the puzzle. And if all of the pieces are present, we can do this with high confidence. But if we have missing pieces or fragments, then it becomes hard to put it together. And you can see that when we analyze this peptide without any label in isolation, we are able to see quite a few fragments and have a good score. But when we label that same peptide with a barcode that we made at PTI with the help of our collaborators, we are able to identify more fragments, that is more pieces of the puzzle, so we can determine the sequence with higher confidence. We can better localize post-translational modifications such as phosphorylation, and we can better do de novo sequencing, which I think is very important for us in a number of contexts, for identifying new antigens, for discovering new forms of alternate RNA coding. So we are very excited to be thinking of not only how do we get more data at a lower cost, but how do we improve the quality of the data that we generate. And one thing that I think will be apparent today from our presentations is a deep synergism between the things that we've done. We develop new ways to parallelize, that creates new data structures, new challenges. We also need to have new software to decode that data, to take advantage of that structure, to solve the new challenges that we created when we increase data density. We also need to tailor the biological applications and questions. We need to design our data acquisitions according to the biological questions that we ask. So there's a very deep synergy between these processes. And this is an aspect that is very dear to my heart at PTI, that we're not focusing just on the technology, we're not focusing just on biology. We think that a lot of key fundamental biological questions cannot be answered unless we develop the technology. But which technology, how we develop it, that is informed by the biological questions, by the desire to better understand pathology. And we really put a lot of emphasis on the synergy between these projects to make the whole work better than individual parts could ever work in isolation. And as I stated at the beginning, our primary aim is to advance biomedical research, to accelerate the pace of biomedical research. And we cannot do this alone. Doing it really requires sharing our technology and methods with the broader community. This is a massive, massive ambitious goal. And we realize that we need partners and we need to empower others to use our technology. So we are very passionate to be able to share and support others using the technology that we've developed. I think there are plenty of problems that can benefit from it. And we are very fortunate to be philanthropically funded by visionary supporters who understand the importance of data sharing and support our desire in empowering the community. So we put a lot of effort, not just in tossing a data set, here, you can go figure out what it is, but we put effort in describing what we have done, creating metadata that are useful so that the data can be reused, and also organizing information sharing, such as this symposium today, where we aim to broadly enable the community to use what we have created. And of course, I would like to thank all of our supporters, certainly Convergent Research, that have played an important role in facilitating the formation of PTI, and Eric Schmidt Features, and Ken Griffin for providing funding to support our bold foray into making proteomics more powerful in the service of accelerating biomedical research. Now, before I invite Harrison here to introduce the first speaker of our first session, I do have a request to make from you. We had a wonderful group of people here, very diverse group of people, also many people online who are watching this. And some are leading experts in the technology, some are leading experts in the biology, and there are many people who don't have deep expertise in one or the other. And we want to make these presentations as accessible as possible. So to do that, I need your help. I need you to help ask questions about all the jargon that we might slip into using. I try to avoid that, but the truth is it's hard. There will be some jargon. So please keep us in check. Ask questions of what is not clear, because our goal here is not a showcase. Our goal is communication. and to communicate well, we want to make this symposium as interactive as we possibly can, and this requires your participation. So I'll stop here and invite Harrison to come and introduce the first presenters.